So it's my pleasure to introduce Wan-Ju Lee. Everyone knows Wan-Ju. She got her bachelor's degree at China Medical University in Taiwan, and then got a master's in pharmacoepi at Taiwan National Defense University. That's right. National Defense University?
National Center Defense Center.
National Center Defense Center. Anyway, besides learning pharmacoeconomics, she also learned some fighting skills. So don't piss her off. If you piss her off, she smiles, and then she's apparently able to kill a grown man without you even seeing her move. Like Todd, right now, you can kill him right now. So, also be careful about what questions you ask her, because you might find yourself on the floor. These jokes aren't going over as I thought they would. Anyway, I
don't even know what you're talking about.
I'll stop with the jokes. Sorry. No, well, just a little bit, like a picture of somebody. She's finishing her 4th year of the PhD program. She should be done this summer. She's been a Dean Scholar this last year, which is really an honor. We're really proud of Wan-Ju. She's had a great run here at UIC. She's also getting married this summer. She moved to the U.S. and fell madly in love with Adam. And Adam is from mainland China, so after they get married, she's going to use some of her defense skills.
Excuse me, before this wedding, I need to meet and talk with Adam.
Okay.
Make sure he's worthy.
All right.
So go ahead.
Dr. Schulman, just same for Dr. Schulman, although he wants to make my life a little bit more difficult. But thank you everyone for coming today. Today I'm gonna present part of one of my dissertation projects. It's a risk of infections in children and young adults with JIA or RA who receive TNF inhibitors. So as I said, from my dissertation, I will have 4 papers coming up from this dissertation. The first 2 is basically aiming to looking at the utilization of the TNF inhibitor use, and it's already submitted to the journals and waiting for the reviewers' response. And then today we're gonna focus on the 3rd study, and hopefully I can finish the 4th study by summer. And I would like to present the whole study first, and then so I appreciate if you can save your question at the end. I want to acknowledge my dissertation committee. Without them, I couldn't really go this far, so really appreciate their guidance. All right, so here's my outline for today. Start with children with juvenile idiopathic arthritis. JIA is defined as arthritis in children that persists for 6 months or more in children less than 16 years old. And JIA is also one of the most common chronic disease in children because of the inflammation of the joints and swelling, the pain. So it brings, serious impact about their quality of life. Like children may miss the class, and many of them will carry this disease into their adulthood. On the other side, looking at the incidence of RA, rheumatoid arthritis, in adults. So as we said that some JI can carry their disease into their 20s or 30s. And looking at this graph is the incidence of RA in adults population. We can see that adults aged like 20 to 40, actually the incidence is very low. So even the clinical trial, they are saying that, they recruit patient more than 18, but, like young adults is really underrepresented in the clinical trial. So that's also why we focus on children and also include young adults in our study. This is a treatment guideline for patients with JIA. And if you have a mild JIA, you start with NSAID monotherapy. and or corticosteroids joint injection or just oral corticosteroid use. And if the disease is not treated well, controlled well, then we'll add the DMARD, disease-modifying anti-rheumatoid drugs. And the classic DMARD is methotrexate. And if the patient fails to respond to DMARDs, then they will switch or add on the TNF inhibitors here. And the treatment guideline is similar to adults with RA, like also start with DMARD and step up to TNF inhibitors. So TNF inhibitors, that is actually act on, that will actually focus on a potent cytokine called TNF alpha. So this is a cytokine that could modulate many inflammatory process and the responses in the body. So the design of the TNF alpha inhibitors is try to bind the TNF receptors, so stop the action of TNF alpha, so that it could suppress the downstream inflammatory response of this TNF alpha. Because of the potent efficacy shown in clinical trials, for now, like more than 2 million patients cumulatively use this medication for their rheumatoid diseases or IBD, since they are on the market around 2000. Studies also show that there's an increasing trend of TNF alpha inhibitor use in both JI and RA cohorts. This figures from our first study in our dissertation, also showing that not only this drug is increasingly used, but also the time, like physician team to prescribe this medication like earlier in their disease course. So with the widely use of these biologics, like number of adverse events that was seen in case reports or clinical trials, such as like most common is like infusion reactions, and some also found like serious infections or cancer, especially lymphoma or other different types of adverse events. And today we're gonna focus on serious infection here. And this association between like infection and TNF inhibitors has been already studied in some meta-analysis and also observational studies. So you can see from meta-analysis of clinical trials or observational studies using large claims, we can see the results. It's kind of like inconsistent. Some find an increased risk, like maybe twofold increased risk of infection associated with this drug. However, some study didn't really find that association. But those studies are conducting mainly, like all of them are in adults. For children, the evidence, and not only children but also young adults, the evidence for the long-term safety of this drug is lacking. And we know that children, especially children, are vulnerable. They are more likely to experience adverse event than adults. Their immune system is not mature enough, so they are more likely to have risk to have infections. And from our previous work, we also found that in the FDA adverse event reporting system database, we found like these three, like infliximab, etanercept, adalimumab, they are on the top 20, on the list of top 20 drugs that is most commonly reported with adverse event in children. So when we're looking at whether the evidence for this specific association, there is one systematic review trying to describe the incidence of serious infections in children with this condition, also the type of infection they have. There are only 2 observational studies using the Medicaid database, and then looking at bacterial infection or opportunistic infection. However, they are like underpowered to really find association between this drug and adverse events. So with all that said, the gap is, like we know that it's consistent result there, like mainly in adults, and a few evidence is just conducted in children. And we know children are more likely to having this risk. And because of clinical trial, that's small number, short follow-up period. So, and the current evidence is mostly focused on Medicaid children. So our study is trying to determine and quantify this risk of serious bacterial infection associated with this biologic TNF inhibitors compared to traditional DMARDs in these young groups. Our hypothesis is that TNF inhibitor will have increased risk of infection. And this hypothesis is actually based on the biological plausibility of this medication. So as you can see in the figure that, like when in the body there is a stimulant cell initiate this inflammatory response that will activate a T cell and release a TNF alpha this cytokine. So TNF inhibitors that could, like inhibit the action of that. And although that this level of TNF alpha probably will be more in patients with this rheumatoid disease conditions, but at the same time, like TNF alpha inhibitors may also affect the normal defense system in our body, which is an immune system. So patient may be more likely to have the risk of infection. Here is the DAG directed acyclic graph to showing our exposure TNF inhibitors here, and our outcome is serious infections, and accounting for a number of factors, especially confounders that in our study. And I want to note that, like disease severity is one very important confounder that we need to consider here. It will affect the use of corticosteroid, which is well documented that this drug will increase the risk of infection. But also is that could introduce a confounding by indication here, because patients with RA, the studies show they're more likely to have risk of infection compared to general population. So we want to address this. We don't want to see an increased risk of TNF alpha inhibitor just because patients with more severe conditions of RA or JIA and use this medication, and the risk is not attributed to the drug but the disease. So and also we account for other like patient characteristics or risk factors for infections, etc. In this study, we used Truven market scan commercial claims encounter database from 2009 to 2013, and I'm sure many of you are already familiar with this database. It's a private sector health plan, health data, including patients like commercially insured, and this database provide the identified health data for their enrollment, their healthcare utilization, like inpatient, outpatient, emergency visit, or the medication record they had. My inclusion criteria is during this study period, I will focus on children like less than 16, using these 3 ICD-9 codes to identify patient with JIA. And for young adults, we'll define as patient age 16 to less than 30, and have a diagnosis for RA. And patients have to have at least 2 diagnoses within 1 year or 1 diagnosis by the specialist. And this is based on the validated algorithm. And patient has to receive the new prescription of either DMARDs or TNF inhibitors. And the new prescription is defined as like 6 months. They don't receive any prior prescription of the medication 6 months before the index date, which is the first use of their medication. And the DMARDs and the TNF inhibitors are listed here. DMARDs include methotrexate, sulfasalazine, hydroxychloroquine, and leflunomide. And TNF inhibitors include infliximab, adalimumab, etanercept, golimumab, and certolizumab. Okay. If patient didn't have 6 month continuous enrollment, 6 months before their index, before the new use of medication, they'll be excluded. Or a patient who had TB, tuberculosis, or their medication treated for this disease, or diagnosis for cancer, transplant, or HIV infection will be excluded. We'll start to follow from their index date, which is the new use of medication, until, if they have the infection or they switch the medication, disenroll from the plan, or discontinuation of their medication use, or end of study period. Our study outcome is the serious bacterial infection requiring hospitalizations using the IC9 codes in the inpatient settings. And we only count for the first occurrence of the infection. And here are the codes we use from a study like validate this ICD-9 codes. Our study exposure are either new use of DMARDs or TNF inhibitors, and they're all initiator. And and we want to have a clear follow-up for the percent time that's only contributed by one of the drugs. So if they switch, we'll censor them. And in the baseline, if they couldn't use either or the other medication. So I'll illustrate with this figure. So during the study period, we'll identify patient study cohort with inclusion/exclusion criteria. And then if they have the new use of the medication, then we'll have the index date, which is a T0, start to follow them. And we'll follow them until one of these criteria, censoring criteria. For example, this patient, they start to use DMARDs, and then later they add on the TNF inhibitors in their disease course. Then we'll follow this patient from T0 until they switch. For patient B, if they only use a single medication, but if there's a gap more than 90 days, then we'll stop using that. We want to make sure all the events occur while the patient is still using the medication. For this patient, they have overlap use of two medication later, and then if an infection was found in during this time, but like I said, I will just stop when it switch, so that outcome will not be counted. But for this patient, if the infection occurs during just exposure to only one single drug, then we'll stop when they have that outcome. We account for many study covariates. We measure their baseline demographics, healthcare utilization, and also whether they have the procedure for their RA condition. And also factors related to infection, like number of outpatient, inpatient visits for infections at baseline, also comorbidities or co-medication that would related to affect the immune system of the patients. And in this study, we use this high-dimensional propensity score, which is approach to account for a potential confounding by indication. I mean, in this study, we hope to address that by providing a summary score to predict the probability of receiving the TNF inhibitors in our study. And so basically, it's putting many information into the computer, and that the algorithm to tell us which variables are important to adjust it for. So we put demographic, healthcare utilization information, also inpatient, outpatient diagnosis and procedure using IC9 code, CPT code, and also individual medications. So the algorithm will calculate the bias term of each covariates and then rank that bias term. And then we decide to pick top 500 covariates into our propensity score model to predict the TNF inhibitor use. The patient characteristic will be described in, like mean or frequency in proportion. The crude incidence will be calculated for the infection hospitalizations using number of events per 100% years. We also plot the cumulative incidence function. A Cox proportional hazard model will be used to estimate this risk of infection associated with TNF inhibitors compared to DMARDs. And in this Cox model, we basically, based on the propensity score and based on the distribution, of the prevalence score of these two exposure group, we decided to use as adjustment using tertile of the prevalence score in our final model. And for all the covariates we talked about earlier, it's measured at baseline and then putting into the prevalence score model. But like corticosteroid use is well documented to have increased risk of infection. So if any use of corticosteroid during the follow-up, we're also accounting for that, and we're putting that into our final model as well. Last, we'll do some stratification by age group and gender. So here comes our results. We got like 6,711 patients who initiated their DMARDs during the study period, and then we got 1,442 patients for TNF inhibitors. As you can see that almost 70% of them are actually come from young adults group, and the majority of them are female. For their healthcare realization at baseline, like TNFi group has more outpatient visit and also inpatient visit, as well as more comorbidity calculated by Charlson Comorbidity Index. DMARDs, however, DMARDs have more like lupus or Sjogren's syndrome at baseline, but TNF inhibitors will have more rheumatoid-related condition and also IBD. In terms of their medication use, DMARDs actually at baseline have more corticosteroid or antibiotics use. And the other immunosuppressants actually rarely used in these two groups. And table 2 shows the accrued rates of infection hospitalizations for DMARD and TNF inhibitor groups. So with this number of patients, we got this number of person year. The average follow-up for DMARD is around like 7 to 8 months, and for TNF inhibitors, it's longer, like 9 months. We got not many, total 107 cases. Here is the accrued rates for infection in each group, and we also break down to age group. So we can see that TNFi seems to have higher accrued rates of infection compared to DMARDs. And we can also see this reflect by our accrued cumulative incidence function here. On the top panel A is the accrued incidence rate, like cumulative hazard estimates for overall cohort. The log-rank test shows a significance showing these two curves. So the red curve is a TNFi group, and the blue one is DMARDs. And the x-axis is the time from their medication use until their sensor have the events. And the y-axis is the cumulative hazard. When we break down into children and young adults, like in panel B and C, we can see in children, it seems the curve seems to be like have more separate. And in panel, like in young adults, it's kind of like some areas overlapped and not showing the significance there. And looking into this one or around 100 cases and to see what type of infection they are having. And by organ system, we can see that TNF inhibitors seems to have a higher proportion of skin-related infections, while DMARDs have more respiratory tract infections. When we look into detail, like specific infection, what's the infection they have? So for TNF inhibitors, they have more cellulitis, abdominal abscess. As for DMARDs, they have more pneumonia and pyelonephritis.
Are these statistical comparisons, or are you just
No. It's
so
because
how
it's.
Can you say that there's more? Like,
I mean, just by looking at the proportion, I'm not. Yeah, I probably shouldn't say there's more. I was just looking at the proportion, but I didn't really do the statistical testing since it's the
if they're not
number.
significantly different statistically, you can't say that there's any difference.
Okay, I see. So I will just say the distribution of description of the infection types here. And table 4 shows our main result from our Cox proportional Hazard model. So we found a 60% increased risk of infection, bacterial infection associated with this TNF biologics compared to the traditional DMARDs, although it's kind of borderline significant. The p-value is almost 0.05. However, when we stratify by different age group, and in children with JIA less than 16, we can see this risk is like even higher. It's almost 2.7 increased risk in children. But in young adults, we didn't really see this association. When stratified by gender, we'll see male seems to have like higher, based on point estimate, it's higher than, female, but again, that these two estimates not significant statistically. So our study find a modest increased risk of bacterial infection associated with TNF inhibitors compared to DMARDs in this young population. Especially when we stratify, we found a even higher risk in children with JIA. And in the specific type of infection, we found that cellulitis were most commonly seen in in TNF inhibitors, while pneumonia was more commonly seen in DMARDs group. Our study have some limitations. First, misclassification could be possible, in terms of their disease status and outcomes. However, we used a validated algorithm to identify in the claims data, and even this misclassification exists. It should not be differential between the two exposure group. So that will non-differential misclassification may lead our result toward to the null. Although we used a high dimensional prevalence score to adjust for the confounding by indication, but we can only measure the measurable covariance in our study. So there could be some residual confounding effect in our study, such as associated clinical parameters that are related to the disease activity. So FDA actually issued a black box warning in 2008 in terms of the serious infection associated with medication. So if physician is prescribing will be affected by this warning, they may prescribe this medication to patient less likely to have infection. So if this exists, then our findings actually underestimated true risk. On the other side, the information publication or this warning that come out during our study period. So we're doing the provincial model using the baseline, but because of this publication warning may change the behavior of prescribing. So time varying prevalence score approach might be a better approach to do. So in conclusion, I just want to conclude with in our study, we did find an increased risk for this medication compared to the other immunosuppressant DMARDs, especially in children who have, younger and then who have JIA condition. Our next step is to to go further into the cumulative incidence function to adjust it for the high dimensional prevalence score, and also doing a time variant. So for now, the corticosteroid used during follow-up is dichotomized, and we would like to do time varying covariates for the corticosteroid use during the follow-up. And that's my presentation. Yeah, that's it. So.